All right, this is Notes 6.2 on cell division, okay, also known as mitosis here. And we're going to go through this and talk a little bit about why cells divide. So this is February 2021. You can write your name up top here and the period that you are in as well on your notes, okay? So Notes 6.2. Cell division. All right, so your cells are constantly dividing in your body. We talked about cells in Unit 3. You know that you are made up of cells. That's the basic unit of life. And in order for you to do several different things, you need to divide your cells. Okay, so one cell is going to divide into two. Those two are going to divide into 4, and so on and so forth. Okay, so there's several reasons why, and you can go ahead and write these down. One reason is that you need to grow. Okay, so the amount of cells that you have in your body right now, that's a lot more cells than you had when you were born. So where did those cells come from? Well, remember when we talked about cell theory, that cells can only come from pre-existing cells. Cells just can't pop out of nowhere. So cells are going to come from previous cells, and the way they do that is by dividing into new cells. And so in order for you to grow, you need to divide your cells, okay? You need to replace dead cells. So you have dead cells, you have cells dying constantly, okay? If you've ever looked in a light and maybe you've taken your arm and you've just kind of rubbed your hand or your arm in the sunlight, you will notice all sorts of stuff coming off of your arm, okay? Those are dead cells. So dead cells are constantly being shed off of you. Every time you brush up against skin, you are brushing off tons of dead cells. So if you were not replacing those cells, you would just disintegrate into thin air over time. And so we need to replace those cells, and those cells are replaced by new cells that come from this process of division. Okay. Now, another reason why is that you need to split up cells that are getting too big to maintain. And so if you have a cell and it's growing and it's getting larger and it's larger, at some point it's going to be really large to the point that you can't maintain it. Okay, just like if a city continues to get bigger and bigger and bigger and bigger, you are going to need to divide that city up into 2 cities because the government will not be able to manage the entire thing. All right, so same thing with cells. We're splitting up the cells because it's too big. It's better just to have 2 cells than 1, okay? Keep in mind when we talk about replacing dead cells, this is part of the healing process. So if I cut myself and I injure myself, I lost skin and I lost cells that are now dying off somewhere. Or if I get scratched, okay, I need new cells to replace that area. So in order to grow, heal, replace dead cells, and split up cells that are getting too big to maintain, I need to do this process of cellular division, which is also known as mitosis. Remember, osis is one of our words that means process, okay? So as one cell divides to become 2 new cells, the 2 new cells are called daughter cells, okay? So daughter cells would be the 2 cells that came from the 1, okay? In order to ensure that every new daughter cell has the same copy of the genome, because we want the daughter cells to have identical genes to the original one, or else you would be having a new genome every time you have new cells, okay? The cell needs to go through mitosis. Now, before mitosis can happen, you have to duplicate the entire genome. So this is also known as replication or copying or duplication. You can use any of those words. So kind of think of it like this. Let's say that you are a librarian and your library is getting really big, and at the same time you have way more people. The city's getting super big, and so you have a lot of people coming into your library, and soon enough you realize your library is too small to hold all of these people. All these people want to come in, they want to read books, they want to rent out the same books, and there's no way that you can keep running it like this, okay? So let's say that you split your library into 2. you're going to have the Elkins Library 1 on one side of the city, and then maybe we're going to, when we split the city into 2, we're going to build a second Elkins Library, okay? So we have the second library over here. Now, I want my second library to be identical to the first. So I'm going to have to make a copy of each of my books. Now, before we split, the copy of each book is going to be attached to the original book. So I know where they are, and then I can split them up later, okay? So same thing happens. Remember, we talk a lot about chromosomes kind of being like books or recipe books, and then the genes inside of them being like individual recipes. So we need to duplicate this. Kind of imagine this being a book right here, and then we duplicate that into 2 books. All right, they're going to attach to one another. So you can label this right here. This would be what an unduplicated chromosome would look like. So if we had a chromosome and there was no duplication, like it's just a single book, we didn't duplicate it, we didn't replicate it, we didn't copy it, duplicate it, okay? It would just kind of look like this. But through the process of replication, okay, and you can write the word replication here. So you're going to label this blue chromosome here unduplicated, and then we're going to replicate it. We're going to form a duplicated chromosome. We still consider it one chromosome or one book while it's still attached, but when we detach them, that's when they're going to be considered 2 separate chromosomes. We'll look at that in a bit. So this is kind of what chromosomes look like, and you know that's what chromosomes look like because you've seen those X-shaped shapes, all right? So one side is just one copy and the other side is the other copy, and they're kind of stapled together in the middle in a sense, right? Okay. So this is, let's say this is paternal chromosome 2, okay? You would have maternal chromosome 2. I would color that maybe red or whatever. This is not 2 chromosomes, and this is not the maternal and not the paternal together. This is just one copy 1 of the paternal chromosome, and then copy 2 of the paternal chromosome. And then you'd go on to chromosome 3 and chromosome 4 and chromosome 5, and you do the same thing for all of those. All right, so after you've labeled that, let's go down here, and as we explore mitosis, we're going to use this color-coded cell with these shapes here to help us explore how a cell divides and what that process looks like. So the first thing that you need to know is when we look at this cell, let's label this right down here, the cell membrane. So my little black line, my black circle right here, that is the cell membrane, which holds all of these things together. And this brown thing right here is what we call the nuclear membrane. So that's just the nucleus. Remember, the nucleus contains all the DNA. It is the library. So we have the cellular membrane and then the nuclear membrane inside of the cell. Now, there are some new terms that we're going to learn. You'll see these little T-shaped organelles in a sense, all right? Those are called centrioles. They're kind of along the center line of the cell. And what they do is they make what we call spindles. Okay, so centrioles make spindles. Spindles are kind of like ropes that the centrioles are going to use to move around the books or move around the chromosomes in the library. So the chromosomes, remember, we want to pack up our DNA and our recipes inside of these tightly packed bundles so that we can move them easily. And what's doing the moving? The spindles are doing the moving of those chromosomes. So the centrioles form the spindles. The spindles are made out of microtubules. You learned about microtubules in 3.2 when we talked about cellular organelles. Remember, microtubules are just these protein railways, okay? Here they're kind of used like ropes. After you've labeled that, we're going to be using some color coding of these different chromosomes. Now, normally, a human chromosome has 23 pairs. That's a lot to draw. We're not going to draw all 23 pairs. We're just going to draw 2, but just know that this process happens with all 23 pairs. So let's label these chromosome pair 1. Okay. You can tell that they're a pair because they're the same size. But one is maternal and one is paternal. So we're going to label this maternal chromosome 1. Note how it is duplicated. So you have one copy of maternal chromosome 1 and another copy of maternal chromosome 1, and they're kind of attached. That's in a pair, and that is paired up with paternal chromosome 1. So again, this is your father's version of all these genes, and this is your mother's version of all of these genes, and you have both of them, and you want both copies when you divide your cells. Okay, so that's like chromosome pair 1. Let's look at chromosome pair 2. So these are both smaller in size. They contain fewer genes, so it's just a different bundle of genes. They're smaller, okay, and you have a maternal version of that bundle, and you have a paternal version of that bundle. So go ahead and label these. If you want to color code, use red and blue if you want, you can. But you've got the maternal chromosome and the paternal chromosome 1, maternal chromosome 2, and paternal chromosome 2. Keep in mind, you need to understand and wrap your mind around these chromosome pairs and duplicated chromosomes and chromosome 1, chromosome 2, chromosome 3, chromosome 4, maternal and paternal. Sometimes that can be a little bit confusing, so if you just need to take the time to write or draw down your own little diagram, you do that. Do what's helpful for you. Hopefully the homework will help you with that, but I will be asking questions about this kind of stuff on quizzes and stuff. So you just need to know how your DNA is organized in your cells. All right. If you flip, and you can pause at any time that I'm going too fast, you flip over, we're going to look at the steps of mitosis, okay? So mitosis is Greek for a thread process, okay? Mite is a thread and osis is a process. It's kind of the process of using threads or little strings to organize your chromosomes so that you can divide the cell. So the purpose is to divide a normal body cell into identical daughter cells. What do I mean by normal body cell? It just means I'm talking about like skin cells or cells in your eye, cells in your ear, cells in your liver, that kind of stuff. I'm not talking about sperm cells or egg cells. So anytime I say normal body cell, I'm just not talking about sperm and egg. We're going to learn about those later, but this is just a normal cell, okay? It does this whole division process. If we're going to create new cells and we divide, it does this whole process in 4 steps, which I'm going to call PMAT. Isn't that lovely? Okay, PMAT. That's because we have 4 phases. One starts with a P, the next one starts with an M, the next one starts with an A, and the next one starts with a P. Okay? The time that this process takes really depends on the cell. So it can take anywhere from minutes to hours to days. And so a typical skin cell might divide in less than a day, but then in your liver, it might take a whole year for it to undergo mitosis. And so, yeah, it's crazy the amount, just depending on what kind of cells you have, where they are, their division process can take a long time. And yes, just remember we're showing 4 chromosomes, not all 46. We're showing 2 pairs, not 23 pairs. All right, so I'm going to stop sharing this slide, this Word document, because I'm going to go through a PowerPoint animation of this process, okay? So this is the process of one cell dividing into 2 cells, and I'm going to show up here what the steps are, and you can follow along by filling in the blanks when we get to that, all right? So let's go ahead and present. We're going to go through this process here. All right, I just wanted to show you a couple of animated animations and videos showing this process happening. Okay, so here is a scan of a cell. The time is in hours, and so this takes about a whole day for it to complete, about 20 hours for this particular cell. Notice it's 10 micrometers long about, so micrometer, remember the mu in the micro there. So a millionth of a meter. Here the chromosomes are orange, and so you'll see them lined up in the center and then spreading out to divide into two cells. So we had one cell at first, and then we had two cells at the end. So where there once was one, there now is two, and we've divided. All right, here's another animation or a scan. This is actual scans of what's happening inside of this one cell. You'll notice the spindles are those long stringy things, and they are grabbing onto the chromosomes in the middle, and they're coming out of the centrioles on the side. Okay, here's another very sped up animation. So here's a cell, and zoop, two cells. One cell, and then zoop, two cells. All right, so you have two cells where you once had one. That's great, you're growing. Okay, and then those cells are going to grow, and then they're going to divide. Remember, if you just have one cell here, and it takes those chromosomes and it divides it up, and then you have two cells, then that one, then each of those two cells can then start dividing on their own. So then you would get four, and each of those four would divide, and yeah, so on and so forth. Okay. So here's our first diagram. This is your cell, just an average cell. We're only showing 4 of the chromosomes. We've already labeled this, okay? So before mitosis happens, before cellular division happens, a normal cell looks like this. Its DNA is just kind of unwound. It's floating in the nucleus. It's not really tightened. It's not, the chromosomes are not tightened up yet. They're kind of unraveled a little bit, and they're just kind of floating around in the nucleus there. You still have those individual stringy molecules that come from the mother and the father. But what starts happening is the DNA molecules start wrapping tightly into their duplicated chromosome shape. And this is the first step of step 1, prophase. So go ahead and fill in prophase as step 1 on your notes. So step 1 is prophase. So the DNA molecules wrap tightly into their duplicated chromosome shape. So they've already duplicated. Now I've got 2 copies of each one stuck together. And what starts happening is these centrioles, the green T-shaped things, start building the spindles, which, remember, that's just the microtubule system. So again, fill this in on your notes. The centrioles start building the spindles, and those ropes are going to be used to bring these chromosomes into their proper place. Okay, the nuclear membrane starts breaking down. So you'll notice here it used to be solid, and then now we're starting to dissolve the nucleus because we need to get those books out of the library in order to move them. So we're breaking down the library. We're breaking down the nucleus, okay? Now here's a way that you can remember this step. Pro, remember pro means before. So this is like the before we're actually dividing. I mean, we are still in the process of dividing, but it's before the other steps. And so we say prophase is the first step. So that's how you can remember the first P in PMAP, okay? If you need to pause and write down these terms, you can. All right, let's go on to the next step. The next step is called metaphase, okay? So metaphase is when I have lined up my chromosomes in the middle. So the duplicated chromosomes line up, so they've gone from the nucleus and they've started lining up to the middle of the cell. So duplicated chromosomes line up across the middle of the cell. What starts happening is spindles connect to the center of each chromosome. So these spindles are being built by the centrioles. They were kind of already built in prophase, but now they're actually connecting to the center of the chromosomes. Right? Here's the way you can remember this. Meta kind of sounds like middle, so you can kind of think of the middle phase. And notice where the chromosomes lined up. They're lined up in the middle. Okay. Ooh, what just happened here? All right, so something's going to happen to these chromosomes because Okay, these are, remember the two copies are together and we're gonna split those copies. All right, so now they've just been split and we're prepping to make our two new cells. And as those chromosomes move apart, we call that anaphase. Okay, anaphase is the third step. The spindles are separating those duplicates and they pull those chromosomes to opposite ends of the cell. Okay, so the spindles separate the duplicates. Spindles separate the duplicates and pull them to the opposite ends of the cell. Okay, this is how I like to remember anaphase. You'll notice the chromosomes kind of look like A's on their side. So the letter A without the little bar in between. Anytime that you see a picture or a diagram where the chromosomes look like that and they're moving across the cell, you know it's anaphase because they're shaped like little A's. Okay. Keep in mind, this little blue guy is still one chromosome. So it doesn't look like an X, but that's fine. Right now, I've got 8 chromosomes. Before, here I had 4 chromosomes. It's kind of weird, right? 2 copies of 4 chromosomes, so 4 duplicated chromosomes. And right here, I've got 8 chromosomes, and they're identical copies of each other. Okay? So in the human cell, it would be more than this. Okay? All right, so here's what happens. The cell starts to elongate, and we're going to start splitting and kind of pinching in the middle here. So we've gone from here to, we're going to start pinching here, and you'll notice that the spindles can kind of let go of the chromosomes. And then we're going to move into telophase. Okay, so telophase, we're starting to build those 2 new cells. So let's look at what telophase is. Chromosomes at the opposite ends. Now that they're at the opposite ends, they can start to unravel, so they don't need to keep their shape. Okay, the nuclear membrane's starting to form again, so we've got 2 new cells, 2 new cities, and we're going to start building those 2 new libraries. Okay, so our daughter cells are coming. We're about to split off. So chromosomes at the opposite ends start to unravel, nuclear membranes start to form again. I'll wait for you to write that down. And then we have, so that was the last step of mitosis, and then we have what's called cytokinesis. Cytokinesis is not necessarily a part of mitosis. It's more like right at the end of mitosis where we've created already 2 new daughter cells. So you have the process of division or mitosis, and then cytokinesis is just that last part where now I've created 2 new daughter cells. I used to have 1 cell, now I have 2. All right, so after mitosis happens cytokinesis. Remember, cyto means cell, kinesis means movement, is means a process. So this is the process of cell movement, particularly of cytoplasm movement. The cytoplasm kind of pinches in half, and once it pitches in half, you've got 2 new cells, and they're identical cells called daughter cells. Notice each one has a paternal and maternal chromosome 1 and a paternal and maternal chromosome 2. That's great. Okay, you'd have 23 pairs in each one. Here we just have 2 pairs in each one for the picture. All right, so 2 new identical cells. So there's another picture of our daughter cells. Okay, 2 daughters where there once was 1 cell. Now, let's say that these cells need to divide again. So if we want to make 4, okay, if we want to make out of this, all right, and they're going to just do mitosis again, right, it would need to duplicate those chromosomes. So here, this is unduplicated, that is duplicated, and then we can start the whole process of mitosis again. But we're not going to consider that. We're just done with mitosis at this point. Okay, and if they're not going to divide, they're just going to unravel that DNA, and you're going to have this kind of tangled mess that somehow the cell still knows how to read, still knows how to obtain those recipes. Okay. Now, this is really important applications. When mitosis goes out of control, it causes cancer. Okay? It causes cancerous tumors. That is what cancer is. Cancer is mitosis gone rampant, gone unregulated. Okay? It's when cells just start dividing like crazy and nothing's keeping them in check, nothing's slowing them down, and you get a tumor, which is just a large collection of new cells that are dividing and dividing and dividing and dividing. And if that gets into your bloodstream and that travels somewhere where it shouldn't be, that's cancer, that's dangerous. Okay, that's malignant. So mitosis, understanding how cellular division works, okay, if you're passionate about finding better treatments for cancer, okay, understanding mitosis is a super important part of that process. Now let's just think about this as a thought experiment. Why is it important that we duplicate the chromosomes? Why do we need to make the copy? Well, what if the chromosomes had never been duplicated? If they've never been duplicated, let's just consider this. Here's a this is not real, but this is our thought experiment. Here's a cell, and it did not duplicate its chromosomes. So you'll just notice that the chromosomes are just there. They don't have any copies attached to them. They're half of an X-looking thing. Okay. And let's say I get over to metaphase, and my chromosomes kind of line up in the middle there. Well, I need two new cells, and they need to be identical, where I need a paternal and a maternal version of each chromosome. And so when I start pulling them apart, if I only have one copy or one duplicate of each one, I mean, how's it going to choose which one it's going to do? And so then it would create two cells that look like this. Okay, and those are not identical. The daughter cell 1 only has the maternal chromosomes. The daughter cell 2 only has the paternal chromosomes. And we need both. And so if this was the original cell, the daughter cells would not be equal if we did not duplicate. So that's why it's important to duplicate your chromosomes, because if you make a copy, one of those copies can go to one of the new daughter cells, and then you'll have identical cells. So that's why we need to duplicate. Let's look at a recap animation of what's happening. Okay, we've got one cell, the chromosomes line up in the middle, the duplicates split off, and we create two new daughter cells. Those nuclear envelopes, the nuclear membranes reform. Okay, and let's say that it needs to divide again. And so we're going to dissolve that membrane. We're going to line up those duplicates again. We're going to split them off, and we're going to form two new daughter cells. Now, let's say one of those other daughter cells flies off, and then maybe it dies, and you need to form another chromosome or another cell. So then that cell again. Okay, notice we're just forming new cells, more and more new cells. It's going to grow exponentially. And that's how you grow, and that's how you create new cells, and the process just keeps on going. Yeah, and that's crazy for you to think about how all the cells in your body kind of went through this process. Most of the cells in your body went through this process to come from your single cell when you were an egg that was fertilized. Okay, you went from that mitosis, divided into two cells, divided into four, each of those four divided into two, and so on and so forth, 16, 32, you get all these different numbers of cells, right? That's how you grow from one single cell, is this whole process, and that's why each of your cells has an identical copy of your genome and your chromosomes. Okay? There's just another. All right, so we are done with that. That is what cellular division looks like. That's how it works. It's the process called mitosis. When it goes wrong, that's called cancer. All right, you can work on your homework at any point now. You'll get a chance to do that tomorrow, and you can also start early today. You will have a quiz 6.2 on mitosis, and you will need to know a lot of these things. You'll need to know the phases. So have memorized prophase, metaphase, anaphase, telophase. You need those words memorized. If you want to draw flashcards or something to help you with that, you can. You'll get an opportunity on the homework to do that as well. But yes, so do your homework to practice. There's mitosis. That is how the process of cellular division works. If you have questions, let me know.